Hey everyone, Lauren from BioWay here. This is a Unit 2 PowerPoint review for AP Biology, Cell Structure and Function. If you like this resource, the same PowerPoint can be found at my store on Teachers Pay Teachers. This video is part 1 of Unit 2, which covers the following topics: cell structure and function, cell size, and compartmentalization. Let's get started. So, cell types. Going easy, working our way up, right? What is the difference? Biology's a bunch of opposites. Prokaryote, no nucleus, small and simple. And versus eukaryote will have that nucleus. It's going to be large and complex with membrane-bound organelles. And who's who? Who are the prokaryotes? Who are the eukaryotes? Prokaryotes will be bacteria and archaea. Everything else is a eukaryote. Here are the kingdoms of life. So we have kingdom bacteria, archaea, protista, plantae, animalia, and fungi. So archaea are very similar to the bacteria. They're the extreme bacteria. So if you ever come across them, that is the archaea. Kingdom protista is a eukaryote. So they're going to be the unicellular, mostly unicellular microscopic eukaryotes. Very similar to bacteria, but they do have a nucleus and other membrane-bound organelles, such as chloroplasts, like this euglena pictured. Another example of protista would be amoeba, also algae. So kingdoms of life, what biology is all about. Okay, so here are all the cell structures. I'm not going to go in detail with every single one of these. If you'd like me to do another video, just let me know in the comments below. I will touch the main structures, the ones that I think are the most important. But for each of these, I would know the function and what cell has them, how they connect, how they work together, the cell structures. So let's get started. Ribosome, so important. What do you know about ribosomes? You should know that they are for protein synthesis. Protein synthesis. They are in all organisms, so that is going to suggest common ancestry, a really important topic in AP biology. It will be throughout the course, especially the natural selection unit. So common ancestry in all organisms. Ribosomes will differ in the location and the type, depending on if prokaryote or eukaryote. Created this chart. So prokaryotes, they're just going to flow freely in the cytosol. Eukaryotes, they'll flow freely in the cytosol, but they'll also be on that rough ER, so fixed on the rough ER. Types, prokaryotes are going to be a little bit smaller, 70S, and then eukaryotes are going to be 80S. In general, they're going to be for protein synthesis. It's pretty much the same mechanism in both prokaryotes and eukaryotes. That mRNA is going to be attached, that ribosome will attach to the mRNA and then make that polypeptide, as we can see in that figure. More on that for Unit 6. And keep in mind, both chloroplasts and mitochondria have ribosomes, and they will be the 70S. Okay, so a collection of membranes inside eukaryotes that work together to transport and package and modify proteins and lipids will be that, yes, endomembrane system. It consists of all these organelles, all of these are structures, and they all contain that membrane. Here we have the nuclear envelope, the membrane around the nucleus. mRNA will exit out of the nucleus and find that ribosome on the rough ER. So rough ERs has ribosomes. So that organelle will be for protein synthesis. After proteins are made, they will be sent out and travel through a vesicle. So vesicles are transport sacs within our cells. They're going to go to the Golgi and they're going to go be modified. Maybe have a carbohydrate attached to the protein, a glycoprotein. Once modified and packaged, the substance will be transported out of the Golgi and go out of the cell. Exocytosis. Maybe it's a protein for another cell. Maybe it's a protein to go on the membrane. So that would be those that I've starred. Lysosomes. Lysosomes are made from the Golgi. They contain special hydrolytic enzymes which digest or get rid of worn out cell parts. So they're kind of like the garbage collectors of the cell, lysosomes. And then we have the plasma membrane, the membrane around all cells. Smoothie R. Smoothie R does not contain ribosomes. It will be important for lipid synthesis, making lipids, and also detoxification. Our liver cells have a lot of smoothie R for detoxification. So that is the endomembrane system. I would be familiar with that. All right, mitochondria for cellular respiration. We're going to provide a way to make ATP through oxidative phosphorylation and the electron transport chain. I would know the structure for the mitochondria. Be familiar with this organelle, really important. It's going to have a double membrane, an outer membrane. The inner membrane is folded, called cristae. The inner folds are called cristae. This organelle is unique because it has its own DNA and ribosomes. So own DNA and ribosomes. The matrix would be that space in the center. Mitochondria, making that ATP, cellular respiration. Chloroplast for photosynthesis. I would be familiar with this organelle as well, very important. It also contains DNA and ribosomes. It also has a double membrane. So we have the outer membrane, inner membrane. A stack of thylakoids will be called a granum. So the green substance structures are thylakoids. The space is going to be that stroma. Call it like the fluid of the chloroplast, the stroma. Stroma and thylakoids are going to be important because that will be the location for the reactions of photosynthesis. We have the light-dependent reactions in the thylakoid and the light-independent or Calvin cycle reactions in the stroma. More about that in Unit 3. Cell walls, another important structure. Pretty much organisms have cell walls except for animal cells. Bacteria and archaea have cell walls, plants have cell walls made of cellulose, and fungi have cell walls made of chitin. Cellulose and chitin, remember those are those structural polysaccharides. And cell walls provide support, shape for that cell. All right, so there are the cell structures. Again, if you'd like me to do another video in more detail of each of these, I'd be happy to. Let's label some cells. That's the fun part of AP Bio, right? Yes. Here we have the prokaryote, bacteria, archaea. Capsule is on the outside for extra protection. Cell wall will be in the middle. Membrane will be touching the cytoplasm, so that green structure is the membrane. Ribosomes are the dots in the cell. DNA is going to be this floating material right there. And here we have a plasmid, which is an extra piece of DNA. So bacteria can have plasmids, extra pieces of DNA, which can be used for genetic engineering. So extra piece of DNA, they're circular. And the DNA in prokaryotes is circular. It's going to be circular and singular. Whereas the eukaryotes, our DNA is multiple and linear. So that's an important difference between prokaryote and eukaryote genetic material. So plasmid, flagella is going to be a tail for movement that some cells have. Some eukaryotes have flagellas. So, all right, so that's the prokaryote. And what type of cell is this? Eukaryotic animal, yes, it's complex. There's no cell wall, there's no chloroplast. It's kind of more irregular, round shape. And I would know those structures as well. And what about this cell? Yes, eukaryotic plant. Plants are usually rectangular or square. We have that cell wall. Plants are going to have that central vacuole. So this really large structure will be the vacuole. We have the chloroplast right here. They're a little bit larger than the mitochondria right there. And we have that endomembrane system, the nucleus, the rough ER, the smooth ER. Golgi will be right here. So ribosomes, most likely the dots in the cells in that cytoplasm. So that was the quick plant cell structures. All right, so that's the animal cell, plant cell, and what about this cell? Looks like an animal cell. But what if that outer structure is a cell wall? Then it wouldn't be an animal cell. It would be a fungal cell. Yes, so eukaryotic fungal cell, very similar to the animal cell, but has the cell wall. And something to keep in mind, mushrooms, yeast, they don't have chloroplasts, so they are heterotrophs, consumers, specifically decomposers, those mushrooms. All right, here we have 2.9, compartmentalization. It's a big word, but it basically means how the organelles provide different compartments for the cell. Each cell has, or each organelle in the cell has its own job. For example, the mitochondria, it's for cellular respiration, has a lot of membrane folding, which increases that surface area. We have the chloroplast, the stacking of the thylakoids with that job of photosynthesis. The nucleus contains that genetic material. Transcription is gonna occur in the nucleus. It's separated by that membrane. We have the smooth ER, the rough ER. The rough ER has those ribosomes. So it's a really big word, but it basically means just those organelles have a different function. They can work together, but they're separated by that membrane. All right. And 2.10, origins. How did it come about, that cell compartmentalization? Endosymbiotic theory, an important theory to be familiar with. It's basically how some prokaryotes got engulfed and later became that eukaryotic cell. So we had an aerobic prokaryote, a photosynthetic prokaryote, which later became the mitochondrion chloroplast. And there we have that eukaryotic cell. Long, long time ago, I would be familiar with the evidence. What's the evidence? How are chloroplasts and mitochondria similar to those prokaryotes? Well, pause the video if you want to try to answer it yourself. So chloroplasts and mitochondria, they're going to contain their own DNA and ribosomes. The other organelles don't have that. Chloroplasts and mitochondria reproduce similarly like bacteria, a process called binary fission. So they reproduce independently. Chloroplasts and mitochondria have a double membrane, which is kind of evidence of that engulfing. So it did get engulfed into that cell. So that would be the evidence for the endosymbiotic theory. All right. Let's get into cell size. Cell size, topic 2.2, 2.2, surface area to volume ratios. So something good to remember, small is actually better for biology. The smaller, the better. Smaller cells have a greater surface area to volume ratio, and this is a good thing. That means that cell can transport materials efficiently. It can get reactions done. It can go from one end to the other, no problem. So smaller the better for smaller cells. This will be on the formula sheet, these calculations, so be familiar with those, how to calculate that. If given that cell, the specific shape of the cell, what you would do is you'd find the surface area of the cell or the shape, and then you would divide surface area to volume to get that ratio. This is a good example showing that smaller cell having a surface area volume ratio of 6 versus that larger cell, the larger cube, having a surface area to volume ratio of only 1.2. So smaller, better, smaller, better. All right, explain what happens to a cell's surface area to volume ratio as the cell increases in size. What happens? What happens? The surface area to volume ratio is going to decrease. So the smaller cell, the A, will have that higher surface area to volume ratio. So more efficient. Okay, talking about metabolism and organism size. Metabolism is basically all the chemical reactions in our cells. Our cells are busy. Metabolism is mainly on unit 3, but it's also with this unit. So what's going on here? What's going on in this figure? This is what AP Biology is all about, being able to read graphs, figures. So as we increase an organism's mass, what happens to the metabolic rate? It decreases. Yes, we would call that an indirect relationship. So the mouse is going to have a higher metabolic rate than the elephant. It's going to have a higher metabolic rate. And this is the same for endotherms and ectotherms. But why? Why would smaller organisms have a higher metabolic rate? Scientists are curious. Well, let's think back to that cell. That smaller cell had the higher surface area volume ratio. Kind of the smaller organism has that higher surface area volume ratio. A lot of reactions are happening really fast in that mouse. He's going to have heat loss. He's going to have to make up for that. So they have to metabolize faster, reactions. So in general, that is the trend. As we get larger for an organism, that metabolic rate will decrease. Oh, well, let's make sure we are good there. Here's a practice question. As an organism gets larger, what happens? A, B, C, or D? Yes, it is B. Surface area to volume ratio decreases and metabolic rate decreases. But cells and organisms, even though, humans are pretty large, we have ways to maximize that surface area, and organelles do as well. So I would know kind of some examples. We have the folds of the mitochondria, folds of the ER, the Golgi, so that would maximize surface area. We have the stacking of the thylakoids and the chloroplast. And we also have folds in the small intestine, our microvilli. This allows for more absorption of those nutrients for digestion. So that would be ways to maximize that surface area. All right. So that was 4 topics for AP Biology, 2.1, 2.2, 2.9, 2.10. That would be part of Unit 2. I hope that was helpful, and I'll see you next time.